comprehensive training materials on using the Phage genome annotation pipeline, and are also making educational video modules to go with the developing training material. So today, the format of today's webinar is a little different. Jason has planned to stream their first training video out of a series today, and then he'll be available for the question answers. So once the video is completed, then we'll have the question answer session as usual with Jessica Sacher from Phage Directory. So with that brief introduction, thank you, Jason, very much for your time, and you may please start.
Okay, so you can hear me okay?
Yes.
All right, great. So thanks for the introduction. Thanks for the invitation to present here today. So my name is Jason Gill. And rather than doing a live talk, what we decided to do is to stream basically some of the training videos that we have just finished creating, which describe our Galaxy and Apollo based Phage annotation platform. So we've been meaning to make these videos for a little while, and this kind of spurred us to actually get them finished. So this is actually the first 3 videos in a series that will basically introduce the concepts of the Apollo Galaxy system and just give an overview. This is not going to give you a lot of like step-by-step, how to annotate a phage in here. It's an overview of the system. At the end of the video, and also the links that will be made available later, there are links to training material, which have more explicit instructions on how to, mechanically, like how to upload files and what buttons to press to have things work on the system. So this is the world premiere of this video. So I am just going to basically hit play, and then I'm going to turn my camera off, and this will go. And then when it's done, I will turn my camera back on, and then we can have a Q&A afterwards. And hopefully this does not freeze up or die in the process. Okay, so I want to do this. I'm going to turn off my camera to save bandwidth, and I'm going to hit play on here.
Hi, my name is Jason Gill, and I'm a member of the Center for Phage Technology at Texas A&M University. In this video, we're going to talk about the Center for Phage Technology's Galaxy and Apollo platform for whole phage genome annotation and analysis. First, we'll start off by talking about what is Galaxy. Galaxy is not an analysis tool in itself. Galaxy provides a platform for performing reproducible bioinformatics research. It provides a web browser-based user interface for other command line tools. It provides the history of actions performed and saves your tool outputs. It allows users to chain operations together into workflows to perform very complex analyses. Finally, Galaxy is an open source, meaning free to you, platform with an active user community. Galaxy operates by interfacing with any Linux command line program by a short script, which is called a wrapper. The wrapper represents the input options to the user and passes these properly structured commands back to the program, such as, for example, BLASTP or Glimmer3, or any other command line tool that you may be interested in using. Galaxy runs that job and keeps a record of that job. It then keeps track of the inputs that were sent to it, and it keeps track of the outputs that are produced, and which are all placed in a user history. Ultimately, Galaxy offers the power and flexibility of command line bioinformatics to the average biologist just by using a simple web interface. So why use Galaxy? This is a simple diagram illustrating some of the benefits to the user for using Galaxy. On the left is the user, which is you. And what you see is just the web browser interface for Galaxy, and what you don't have to see or manage are the server clusters, the cloud computing infrastructure, or the complicated command line interface and file management for your tool inputs and outputs, as those are all managed and organized by Galaxy. Why use Galaxy for Phage? Galaxy is a popular and well-supported bioinformatics infrastructure. There are over 125 Galaxy platforms currently running around the world which are publicly accessible. Galaxy automates tasks and retains inputs and outputs for future reference. It's customizable for individual use cases, and the user retains control of the analyses. It has uses in education where students can see and interpret their tool outputs and customize the analyses themselves, which is an advantage over the fully automated annotation platforms that are available. It's useful for novel or unusual genomes, which will permit the user to customize or modify their analysis approaches. It also allows the user to go beyond annotation to conduct comparative genomics, mutational analysis, and phylogenetics. This is a simple overview of the general Galaxy interface. On the left panel, there is a list of tools which are available to the user. The center panel is dominated by the analysis and results. This is where, when invoking new tools, options will appear. And this is where results can be displayed when the tools have finished running. The right panel is the user history, which keeps track of all jobs that are executed by the user in the order that they were run. This is an example of the general order of operations while working in Galaxy. The user uploads data on the right into the history of their Galaxy account. From the history, that data can be passed on to tools in the tool interface in the main window. What the user does not see is the tool interfacing with the wrapper, which invokes the command line tool or script. The output of the tool is then returned to the history. This process can be repeated as many times as needed for different analyses. At any time, the user can then download the results of any individual analyses or data sets to their local computer for analysis on their own machine or elsewhere on the internet. This is an overview of what the history looks like in Galaxy. The history panel contains all input and output for your analysis. All of the input data for tools, such as sequences or GenBank files, have to be uploaded into the history in order for them to be used in Galaxy. Tools in Galaxy can only access data when it is in a data set in the history. All running jobs appear in the history in the order that they were entered. All output data will also appear in the history. Each item in the history is called a data set and appears in the order that it was entered. Each item is numbered. The numbers can't be changed, but the names can be edited by the user if desired. Gray items seen at the top of this window are queued to run, which means that they've been entered by the user but have not yet reached the command line interface yet. They may be waiting for processing time from the server, or they may be waiting for a previous job to finish if they require that tool's output as their input. Yellow items have been passed off to the tool and are currently running. The amount of time it takes for them to run is dependent on the tool which is being executed. Simple scripts might execute in only a few seconds. Certain analyses which are more intensive, like BLAST or interpro scan, might take several hours to complete. Green items are jobs that are completed and ready for viewing. They can be downloaded by the user, or they can be input into the next tool. Red items are jobs that failed to run or returned an error. In the case of red jobs, the user can investigate the bug report which is generated to determine if it's a user error or a problem with the system. The user can create an unlimited number of new histories to keep track of related analyses. The window here shows the history browser window, where the user can sort through and manage all the histories they've generated. Data sets can also be copied or moved between histories, and they can also be copied or shared between users in the same Galaxy instance. Analyzing data in Galaxy. The image on the left shows the input wrapper, and this is the wrapper for the common tool BLAST-P. This wrapper has all the same options that are available to the user in the command line, but in this case they're presented on the web page in a graphical interface. The tool options are determined by the wrapper, but these are all switches that could be entered at the command line. The input data for this tool, like with all other tools, must come from the history, so it must have already been uploaded to the user's Galaxy account and appear there as a dataset before it can be used for other analyses. The output of this analysis will appear in the history when the job is launched, shifting from gray to yellow to green. Workflows. One of Galaxy's most powerful features is the ability to connect jobs into workflows. Some analyses take a long time. Rather than waiting by the computer for one job to finish to execute the next one, the output from one job will automatically be handed off to the next when it finishes. Shown below is a simple workflow. This workflow will take an input genome file, which is in the GFF3 format, that contains the annotations and DNA sequence. The workflow will export the entire genome DNA sequence and run it in BLAST-N. At the same time, it will export all the DNA sequence regions and protein coding genes, translate all of these sequences into protein sequences, and then hand those off to BLAST-P for analysis. Each tool is configured before the workflow starts, so it can be left to run once initiated. The results of each step are saved in the user history and can be retrieved later. Workflows are facilitated by the wide variety of parsers and converters that are available in the CPT Galaxy. Some of these are shown here in this window, for example, tools for exporting protein coding genes and for translating DNA into protein. WebApollo. WebApollo is an interactive genome visualizer that supports collaborative genome annotation. It is an extension of the popular JBrowse genome viewer that allows editing of the genome inside the web interface. It is described as a similar concept to Google Docs, but for genomes. WebApollo maintains user annotations and multiple evidence tracks to guide annotations. The CPT has developed tools that bridge Galaxy into Apollo, which are now seeing uses on our Galaxy installation and on other installations around the world. This illustrates the linkage between Galaxy and Apollo, which is often not that clear to new users. On the left, you have Galaxy, where the user can upload their data and conduct their analyses, and they can, if desired, be downloaded to the user's computer. On the right side, you have Apollo, which is essentially an extension for JBrowse and allows for evidence review and annotation. While each user's account in Galaxy is linked to a parallel account in Apollo, each system maintains its own separate database, Galaxy containing user datasets, and Apollo containing user organisms and annotations as JBrowse instances. What we need is a way to link the analyses conducted in Galaxy to organisms in Apollo, and for that we generated the Galaxy Apollo Bridge. It includes tools that can generate and modify organisms in Apollo and transfer these tool outputs from Galaxy to the Apollo database, where they will then appear as new evidence tracks. It also includes features to retrieve data from Apollo back into Galaxy, where they can be subjected to further analysis. The suite of tools are referred to as the Galaxy Apollo Bridge. These tools are generally used as part of workflows that conduct complex analyses and transfer data to and from Apollo, and are not typically executed by the user as a standalone tool. The user annotations in Apollo appear at the top of the user-created annotations track of display, shown in yellow. This top bar is the only track that is actually editable by the user in Apollo. Evidence tracks, which are in the white field below, are generated by various tools in Galaxy, and they are imported into Apollo via the Galaxy Apollo Bridge. Each organism has only a single user-created annotations track, but it can have an unlimited number of evidence tracks below it, which can display the outputs of any kind of analysis that can be mapped onto a DNA sequence. This schematic shows an overview of what the annotation workflow looks like on the CPT Galaxy Apollo system. An unannotated DNA sequence is uploaded into Galaxy and then subjected to a structural workflow to identify genes. The organism sequence and tool outputs are then transferred to Apollo, where users are able to call genes in the user annotation track based on the evidence. These user annotations are then transferred back into Galaxy, where the called genes are analyzed by additional tools, and these are then transferred back into Apollo, where the user can then use this data to annotate their gene functions, again in the user-created annotations track. When they're finished, the data can be retrieved back into Galaxy for export as either GenBank or GFS3 format files, for additional analysis, or for submission to sequence databases. So part of the process is conducted in Galaxy, which is where the actual data analysis occurs, and then the actual user annotation occurs in Apollo. All of this work is facilitated by transfer steps, which are accomplished by the Galaxy Apollo Bridge. If desired, the user can continue to run other analyses in Galaxy and import these tracks back into Apollo for as many cycles as they desire. The structural annotation workflow. The CPT's phage genome annotation process divides annotation into two major steps. First, structural annotation will predict the locations of protein coding genes, tRNAs, and transcriptional terminators. The functional workflow analyzes the protein coding genes identified during structural annotation to determine their function by a combination of similarity and conserved domain searches. This is an overview of the CPT Galaxy structural annotation workflow. You can see here it's a pretty complicated process. This workflow executes a number of analyses, starting with just your phage DNA sequence to start. By the end of this process, it will have an organism created in Apollo with the structural gene evidence applied to it as tracks, which will then be viewable by the user. I'm just going to go over some of the basic parts here, and we don't need to memorize the entire workflow here. Up here at the top, there are analyses that will identify tRNAs and transcriptional terminators. We currently use tRNA scan and Aridorn to identify tRNAs. We use two different gene calling programs. The first, shown here, is Glimmer3. This is a popular gene calling program and it's one used in NCBI, and this will identify protein coding genes from DNA sequence. As you can see here, the workflow will run Glimmer3 and then process its outputs to be viewable in Apollo. The other structural gene caller that we use is called Metagene Annotator, and again we are taking the tool output and formatting it for display in Apollo. The final structural gene calling part of the workflow here uses a tool called 6-pack. This is what's called a naive reading frame caller. So this will just identify all possible reading frames that code proteins over a certain size. These are then filtered for only those that have a valid start codon, and they get formatted for display in Apollo. The last part of the workflow is the Galaxy Apollo bridge, which will transfer analyses from Galaxy into Apollo, where the user can view them in the context of their genome. This is an overview of what gene prediction tools Glimmer3 and Metagene Annotator will do. What these tools are doing is looking at the DNA sequence and predicting which reading frames actually encode proteins, and they'll display only those reading frames. What's shown here is the output for Metagene Annotator run against the Phage lambda DNA sequence, and above that is a schematic of the Phage lambda genome, which is very well characterized. You can see here that these gene prediction tools are generally quite accurate. This run of Metagene Annotator was able to correctly identify all the known protein coding genes, starting from gene nu1 through gene J, as shown here. It's worth noting that these prediction tools are only looking for normal protein coding genes. They can't find unusual features like, for example, programmed translational frameshifts or embedded genes as you might have with genes in span complexes. But as you can see here, in the case of Lambda, it's done a great job and has accurately identified all the known protein coding genes in the first half of the Lambda genome. So let's give a brief overview of what a prokaryotic gene looks like. A protein coding gene must have a valid start codon, and in our system, valid start codons are ATG, GTG, and GTG. These are by far the three most commonly used start codons in bacteria. The reading frame will encode a protein, and the open reading frame is determined by that start codon. The open reading frame is also commonly referred to as a coding segment, or CDS, which is, for example, how they're stored in GenBank files. At the end of the reading frame, there'll be a stop codon, which terminates the frame. Also upstream of every protein coding sequence, there should be a ribosomal binding site, which initiates translation. So that ribosomal binding site should consist of a Shine-Dalgarno sequence, followed by an appropriate spacer, followed by the valid start codon. The RBS, shown in blue here, has a Shine-Dalgarno sequence, shown here as AGGT, a short spacer, and then the start codon. All protein coding genes must be translated from an mRNA, which requires initiation at a ribosomal binding site. The RBS consists of a Shine-Dalgarno sequence, a 3 to 17 base pair spacer, and a start codon. The Shine-Dalgarno sequence will base pair with a complementary sequence at the 3' end of the 16S RNA to initiate translation of a protein. The strength of that translation is affected by how close the gene Shine-Dalgarno is to the consensus Shine-Dalgarno sequence, which is AGG-AGGT, as shown here in the DNA form. Any subset of that sequence could be used as a potential ribosome binding site with appropriate spacing. So on the right here, the box shows potential valid Shine-Dalgarno sequences. In the cell, translation is initiated from messenger RNA. But since we're looking at this in the context of genome sequences, it's shown here as how it appears in the DNA sequence, since all annotations are conducted on DNA sequence, and DNA sequence is what you're going to be looking at all the time during this process. On the left part of the column here, you can see all the valid Shine-Dalgarno sequences based on the normal Watson-Crick base pairing rules, the consensus Shine-Dalgarno sequence, and also valid subsets that could be used for translational starts. We have to keep in mind, though, that because translation actually occurs at the RNA level, wobble base pairing rules also apply as shown in the right column, where GU base pairs are a valid RNA base pair, allowing some variance in the Shine-Dalgarno sequence. At the CPT, we developed a tool called ShineFind, and we run this tool on all of our structural gene caller outputs. This tool looks upstream of each predicted reading frame, as called by MGA, Glimmer, or 6-pack, and identifies any potential Shine-Dargano sequences, and makes these a part of the gene feature as shown below here. The Shine-Dargano sequence will be appended as a small subfeature upstream of the predicted reading frame. When this gene is called, the Shine-Dargano sequence is part of the gene model, which is called and will appear in the user annotation track in Apollo. A note here on how phage genes tend to be organized. Phages tend to have very high coding densities, typically in the range of 90% or even higher. These features are very tightly packed, and they may actually even overlap by a few codons if that's possible. Phages, because they have to package all their DNA into a head of a fixed size, are somewhat constrained for space, and coding densities tend to be high in phage genomes, much higher than you would see in the average bacterial genome. Phage genes are also typically translated from polycistronic mRNAs, which means they tend to appear in large polycistronic blocks of genes. So we're looking at a gene arrangement here, and in this example, this is the phage lambda genome. You can see the blue and green blocks indicating genes in the plus and minus strand respectively. As you can see here, most genes tend to appear as collinear blocks either on the plus or the minus strand, with very little switching between the plus and minus strands for individual genes. This is an example of output from the structural workflow in Apollo. The outputs shown in these two tracks are from Metagene Annotator and Glimmer3. These will detect most of the protein coding genes in your phage genome. The 6-pack evidence track will show all possible reading frames with a valid start and greater than 25 codons in length. All of these outputs have already been run through ShineFind to add any Shine-Dargano sequences associated with those reading frames. Most of your gene calling should be done from the Metagene Annotator and Glimmer tracks, but 6-pack can be used to identify genes that are missed by those gene callers, or if the gene callers identify a correct reading frame but an incorrect gene start. 6-pack can then be used to identify perhaps the same reading frame with a better gene start. ShineFind helps the user to identify good gene starts by identifying starts that are associated with Shine-Dargano sequences, which the majority of phage genes should be associated with. Another function of the CPT structural annotation workflow is to look for tRNAs. We currently use the tools Aragorn and tRNAscan SE to look for possible tRNA secondary structures. These appear as separate tracks in Apollo when the workflow is complete. As with protein coding genes, it is advised to only call a single tRNA gene at a given position. There's a lot less to interrogate with tRNA calls. It is generally recommended that if it is called by either tool, it should just be called as a tRNA feature. Many phages have no tRNAs at all, especially phage genomes that are less than about 50 kb. So in many cases, the track will simply be empty for your phage genome. Another part of the structural annotation workflow is the identification of row-independent terminators. Row-independent terminators form a stable RNA hairpin that will dislodge the mRNA transcript from the RNA polymerase active site. And these occur at the end of the transcript too, as the name implies, terminate transcription. To find these, we use a tool called Transterm HP. So on the top left here, there's a schematic of what the stem loop looks like, followed by a poly-T tail. On the lower panel on the left, this is a schematic showing what's believed to happen to terminate transcription, where this hairpin snaps shut as it leaves the exit channel of the RNA polymerase, which then causes the transcript to be dislodged from the RNA polymerase active site, which is assisted by the poly-T tail, which has weak base pairing. So what we're looking for here is a short hairpin loop, which is followed by a region of multiple T residues in the DNA sequence. These will also appear in the terminators track in Apollo. So evaluating your terminator output. The Transterm tool provides a large number of false positives that need to be filtered out. It's bad practice to actually annotate every possible terminator that's identified by Transterm. The user really should be looking at this output window shown left for each terminator, which you can bring up just by clicking on the terminator in the evidence track. So we advise that terminators only with scores above 90 are called. Users should look at the 5' and 3' stems. There should be at least 5 base pairs with no mismatches, and the downstream 3' tail should contain at least 4 T's. The user should be very suspicious of low scoring or out of context terminators. This is what we mean by out-of-context terminators. On the top panel, you can see these are all terminators shown as the black box with the arrows, terminating plausible transcripts. For example, they could be directionally in the same register as nearby genes, or they could be located at a position of convergent transcription. Basically, a terminator which is located near the end of what appears to be a transcript are more likely to be valid. In the bottom panel, these are examples of terminators that do not appear to be associated with any actual transcript, and so therefore it is not recommended that these be called. When actually annotating genes in our workflows, we suggest the following order of operations. We suggest that tRNAs be annotated first. This is relatively quick to do, and many phage genomes don't have any tRNA genes at all. It's important because tRNAs may actually occupy part of the DNA sequence that will then exclude protein coding genes from being called there later. The second step is calling protein coding genes, using metagene annotator and glimmer as the main gene callers, supplemented with naive ORF calls from 6-pack. And finally, terminators can be called. Once the structural protein genes have been identified, you have now the context for calling terminators. So the terminators that make sense can then be selected based on your gene calls. The functional annotation workflow. The CPT's phage genome annotation process divides the annotation into two major steps. First, structural annotation will predict the locations of protein coding genes, tRNAs, and transcriptional terminators. The functional workflow analyzes the protein coding genes identified during structural annotation to determine their function by a combination of similarity and conserved domain searches. To begin the functional workflow, we will assume that you've already completed structural workflow and you have already identified all the protein coding genes. The functional part of the workflow will predict the functions of these proteins based on a combination of BLAST, conserved domain searches, and tools including InterProScan, TMHMM, LipoP, and others. The CPT functional annotation workflow is our most complicated workflow. This is a schematic overview of it here. Obviously, we're not going to go into it in great detail. The functional predictions are going to be conducted on protein sequence. DNA sequence similarity degrades rapidly, and amino acid sequences are more constrained by the needs of maintaining protein function. This is why almost all protein functional prediction is based on analysis of protein sequence rather than DNA sequence. The CPT Galaxy functional annotation workflow executes 63 individual processes to analyze phage protein coding genes. In the end, it will produce 18 evidence tracks in Apollo, and these are grouped into 6 track groups, which will appear in the CPT WebApollo platform. The CPT Galaxy functional workflow uses BLAST-P and InterProScan as the major evidence sources for annotation. There are also multiple specialty analyses that are conducted as part of this workflow to identify things like phage spanins, lysis proteins, and genes that are interrupted by introns or programmed translational frameshifts. The outputs of all those workflow components appear after the functional annotation workflow is completed. We're not going to deal with all of those in great detail in this talk. All the evidence tracks that are produced will be displayed in Apollo as shown below. So we're going to be talking mainly about BLAST-P and InterProScan analysis because these are the two main general tools that we use to predict protein function. The CPT Galaxy functional workflow uses BLAST-P against 3 major databases, which appear as 3 separate evidence tracks in Apollo at the completion of the workflow. There is a custom database, which is comprised of RefSeq records from 37 well-studied canonical phages. Some of the most famous phages that are in this database are T4, T5, Lambda, T7, and so on. All of the phages which are in the canonical phage database have been selected because they have some historical significance and at least some level of literature support to support some of their annotations. The functional workflow will also analyze phage proteins against the SwissProt database, which is a high-quality protein database manually curated by EMBL and SIB. And finally, we run BLAST against the NR database, which is constrained only to bacterial viruses by tax ID. So keep in mind that this NR search is not against the entire NR protein database in NCBI. It's against only proteins that are found in any bacterial virus. So this is not a single tax ID constraint. It includes all bacterial viruses, including single-stranded DNA phages and RNA phages. The 3 BLAST results will appear as 3 tracks in the WebApollo interface once the workflow is complete. The other major evidence source for functional annotation in our workflow is conserved domain searching using InterProScan. InterProScan is hosted by EMBL and is integrated into UniProt. Interpro integrates multiple conserved domain databases and assigns a single Interpro ID to related domains. Interpro Scan is a tool that searches a protein sequence against the member databases and detects conserved domains. This is an example of BLAST results in Apollo. The user annotation is in the yellow user-created annotation track at the top. In this case, we have the BLAST NR results in orange below it. Like other evidence tracks, BLAST results will be mapped back to the phage genome and aligned under each gene. The length of that alignment is visualized by the length of the bar below the gene. If the subject sequences are longer than your gene, they will be truncated so they don't overlap with other genes and distort the display. However, if the alignment is shorter, they'll be truncated appropriately. The E-value of your match is roughly visualized by the intensity of the color in the bar underneath. If you mouse over a bar, that will preview the match as shown here, showing a blast hit to a protein identified as a portal protein found in a salmonella phage. Clicking on any one of these individual bars or blast hits will bring up the blast result window for that hit. This includes a lot of information on the quality of that particular hit. The score line is the blast E-value. The lower is better with E-values, and this is also reflected, as I said before, by shading the featured track. BLAST results are automatically filtered to exclude all results with E-values of greater than 10 to the minus 3. The BLAST hit description is also provided. This may be informative and is entirely dependent on the quality of the annotation of the protein record that was used to make the database. This line is pulled directly from the protein entry in the database, so the quality of that description will depend on the quality of the protein record that it was deposited. Also, underlined in red at the top of the window, by the name, is the accession number of the hit protein, which could then be used to go to its record in the appropriate database. You may want to go into UniProt or NCBI to find the protein record and get more information on a given protein. Also of use is the alignment, which is in the gray box in the middle of the window. This shows the match line for the alignment between your sequence, which is the query, and the subject, which is the hit in the database. Obviously, the higher quality alignment here indicates a more reliable blast hit. This is an example of the canonical phage track output from the functional workflow. This is a custom database that contains only paradigm phage with some amount of associated literature. The database includes only 37 phages, so simply mousing over these hits can give a good idea of what type of phage you might have. For example, if you have a large number of hits against phage T4, this would suggest your phage is similar to T4. SwissProt contains the manually curated SwissProt database, and these protein annotations are generally quite reliable as they have all been manually reviewed by staff at SIB or UniProt. So this is an example of InterProScan results in Apollo. Again, the user-created annotations appear in the top, and the InterProScan results are in the white field below. InterProScan searches for conserved domains in 13 member databases. Not all of these domains have been integrated into the InterPro system. The hits here can be used to predict protein function based on conserved domains. It's important to be aware of things like domain swapping, especially if you have a hit to a relatively generic domain like an ATPase domain as part of a much larger protein. Conserved domain hits are more sparse than the BLAST results and often only align to a portion of the protein. Multiple InterPro databases will often find the same or similar domains, but there might be slight variations in how they're named. Again, clicking on any one of the individual InterPro domain hits will bring up a properties window. Not all the databases that are members of InterPro are very feature-rich. Some of the more useful hits when examining this track in Apollo are usually PFAM and TigerFAM, which have more data contained in this properties window. If the domain hit is part of InterPro, there will be a dbxref entry which has an InterPro domain identifier. An easy way to get this is simply to highlight that text in the window with the mouse and right-click on it to search in Google. And that will then bring you to the InterPro entry at the InterPro website. You can then find out more information about the nature of that domain. The calculation of the score for conserved domain searches varies between the databases. This is not strictly an E-value, as you might see it in BLAST results. These tools are all pre-calibrated to show only the results that they think are significant, so you generally do not need to evaluate this value, as you might for a BLAST result. In making a protein annotation, you're generally going to be using either your BLASTP results or conserved domain results, or a combination of the two. When evaluating BLASTP results, it's important to think about what database your hit is in. The preference for databases generally is to use the canonical database or SwissProt databases before moving on to annotations from the NR database, because the NR database has not been curated. You're relying on the skill of other annotators when pulling annotations from that database. And also keep in mind that annotations in GenBank are not generally updated as new literature becomes available. It's also important to think about things like what the e-value is, does the alignment span most or all of the protein between the subject and the query, and also does it make sense that a protein with that function to actually be in your phage. When evaluating conserved domain results, you should think about how specific or meaningful that domain function is. And again, does it make sense for your protein to have that function in your phage? It's also important to make sure that the conserved domain really means what you think it does. You should really go to the interpro record for that domain before assigning a function to your protein based simply on the domain name. Annotating your phage proteins using BLAST and interpro results, you're going to have a mixture of hypothetical proteins and annotatable proteins. Proteins that have no BLAST hits in any databases and have no conserved domain hits will be annotated as hypothetical proteins. If your protein only has BLAST hits to proteins of unknown function and no other conserved domains, this will also be annotated as a hypothetical protein. NCBI is no longer recommending a distinction between hypothetical novel and hypothetical conserved proteins because that status can change when the database is updated. If you have BLAST hits to proteins with annotated function that you believe, or conserved domain hits that you can interpret, then you can annotate that protein with the appropriate function. To add an annotation to a gene in Apollo, in the user-created annotation track, you can right-click on that feature and then click Open Annotation. You can also alt-click on the feature. This will open up the Annotations tab on the right side of your browser window. This tab will show the gene and the mRNA parts of the GFF3 gene models that underlie the Apollo record. You can click on the mRNA field, so the mRNA feature is now active in the Details tab below, as shown by the green arrows. If you enter your new annotation in the name field, you can then click Sync Name with Gene to copy the annotation to both the gene and the mRNA fields. These tabs may take a few seconds to update, so you can click Refresh, or you can click on a different field or tab in the window to make it refresh. What you need to look for here is that the user-created annotations track at the top of the Apollo window, that underneath that gene, your new annotation should appear. If your annotation appears underneath the gene in the user annotations track, that means it's been saved in Apollo. Another feature here is that you can add comments so that you can write some notes to yourself that might support the evidence for your functional annotations. For example, you can have the evidence of BLAST homology to a known protein's conserved domain, or the results of external analyses that you might run, such as HHPRED. You can go to the Comments tab and enter a comment in the box and press the little plus button to add it. You can add an unlimited number of comments to each individual gene. And when the genome is exported, these comments will be exported with the record. Before you submit your record to the public databases, though, you may wish to clear out these comments, particularly if they contain large numbers of personal notes. For more information on using the Galaxy platform, we encourage you to review the extensive Galaxy training resources that are available online. These include those available at the main Use Galaxy homepage, Galaxy 101, and of course the Galaxy training materials produced by the Center for Phage Technology, which are specific to our system, located at cpt.tamu.edu/training materials. The CPT training materials contain detailed instructions on how to upload and manage your genome in Galaxy, and also how to run the major analysis workflows. If you use our system, we also encourage you to cite our publication in PLOS Computational Biology that describes the platform and the associated workflows. The CPT Galaxy Apollo platform is supported by the National Science Foundation, Texas A&M AgriLife Research, and Texas A&M University. We would like to thank the many programmers and scientists who have helped us to design and build the system, and the excellent and supportive Galaxy and Apollo user communities. Thank you.
Okay.
Awesome.
All right.
Thank
so
you. Great. That was awesome.
All right. So it's kind of painful hearing your own recording of your own voice for 35 minutes, but yeah, so I didn't, I saw the questions coming up in the chat. I thought I'd leave it up to the moderators to moderate that part of it. So.
Yep, perfect. I think Ermi, if you're there, you can go ahead and start with the questions that came in on the forum, I think you said.
Yeah. I think there are quite a few questions here, so maybe we take them first. So you may take them first.
Okay, sure. All right, so let me find the first one. Okay, so is the tool restricted or limited to have a proper phage genomic structure like FASTR, or can it be used for gene and protein annotation of phage satellites and genomic islands? Asking for a friend.
Okay, yeah, so I saw that one. So yeah, so the system, it is, the workflows I showed you are really designed around annotating phages, right? And particularly some like the blast searches are really constrained only to phage proteins, because that's assuming all that you're going to annotate. But the system is in a more general sense, like for the gene callers and how those workflows work, they're really good for, and for Shine Find, they're really good for any prokaryotic DNA sequence. They can be used. So you could put a plasmid in there and do the same thing. And one of the advantages here is that you can modify the workflow, right? So the workflows are public, and you can import them into your account, and you can modify those workflows. So you could, for example, change the blast parameter to have it more general, or to target it to a bacterial protein sequence instead, and annotate other things. And one of the things that we are actually hoping to develop with the Apollo people is to have this be a more general, like prokaryotic genomics platform, because Apollo itself it's a very eukaryote-centric system. It was originally developed like for the fly-based people, and for doing complex eukaryote genomes. So we're trying to build in more of the like gene models and features that are specific for prokaryotes.
Got it. Okay, from Varun, if a phage genome is previously annotated, for example, 5 years ago, and has many predicted hypothetical proteins, how much is this tool helpful? Will it predict and annotate all the genes automatically? How efficiently does it work for newly sequenced phage genomes?
Right. So the system was developed, excuse me, the system was developed really for doing de novo genome annotation. So the history really is that we teach a phage genomics course here. It's been running at Texas A&M since like 2001, and we adopted Galaxy in I think 2014 or 2013 or 2014 as a bioinformatics platform to move away from the command line. And so the system kind of grew out of that. And it's really been built around the needs of the class initially, which is that they get unannotated phage genome that they annotate as part of the class. We have a tool right now, which is kind of in a beta state, which will take previously annotated genomes, like for example from GenBank, and import them into Apollo, which means they have to then take the genes which are in that GenBank record and put them in the user annotation track and write them in there, which is a bit of a side use for the Apollo people. So we're working on trying to have that be more functional, in which case then yes, then you can take a previously annotated genome and have it available in there. Otherwise, you could certainly just take the DNA sequence that you have and load it in, and it'll re-annotate, and you have to re-annotate it again. And you probably will get some slightly different results just because the databases are always being updated. And Interpro is being updated all the time. And so if you take a 5 or 10-year-old genome annotation, you probably will find more features if you run it through, say, Interpro scan than you did 5 years ago.
Cool, yeah. How would you compare between PCAN and CPT Galaxy?
Okay, all right, so I mean, I don't want to get political. Right? So, I mean, I've looked at PCAN, and it's a good system also. I think one of the main differences is my understanding of PCAN is it's kind of like a bespoke, like a phage annotation system, whereas ours is on Galaxy. So it is, so we have our own Galaxy instance, for example, right, which runs all these tools, but it is the workflows are modifiable by the user. And the system hosts other tools. Like for example, we also do all of our genome assembly on the system, right? We have tools like PhageTerm running on the system. You can do, we have BWA on the system, so you can do read mapping. So it's a more comprehensive platform than kind of like a very kind of specific annotation system. And I mean, I think that's the big difference. And also because this is Galaxy and open source, what we're hoping is that other Galaxy instances will run these tools. So anybody can spin up a Galaxy server. It's a pretty well supported system as like it's an open source user community. And all these tools, which we have here, are on a public GitHub, and anyone can install them into their own Galaxy instance and run them, and they can have some or all of these tools available.
Got it.
So I think that's probably the big difference.
Yeah. Okay, so suppose that a researcher has gotten his sequence data, forward and reverse sequences in FASTQ files. Are there steps in the Galaxy workflows that could allow him to trim out adapters and primers so he can generate a contig and proceed to downstream analysis, such as phage genome annotation?
Yeah, so our Galaxy platform does have, we have spades on there, right? So we're doing assembly. And we also have like FastQC, and there's Termmatic is on there. So it's all the kind of normal stuff that people would use the command lines. That's one of the nice things with Galaxy, is that any Linux command line tool, if somebody wants to make the wrapper for it, which is just like this XML file that is like presents the options window to you in Galaxy, then it can be executed in Galaxy. So we have made wrappers for a number of tools, but Galaxy has a tool shed, and like a lot of people, like for example, spades. Other people have made wrappers for spades, right? So you just have to have spades in the system, on your system, and then you install it into your Galaxy. So yeah, so there are tools on there for doing kind of all the standard command line, genome, like the read trimming and quality assurance and assembly that you would have on any other system.
Got it. What about phage genome end predictions? Did you?
Yeah, I saw that. So we do have phage term running on the system. So phage term, so there's a lot of things I couldn't cover it all in 37 minutes. So it's so phage term is a tool that looks at read mapping and start and end frequencies. And so it's a command line program. So it is also available on our Galaxy. So to run that though, you need the assembled config that you have, and you need all the original reads from the sequence in order to try to predict the phage termini. And so that it works. It's one of those things that works when it works, and sometimes it doesn't work. It depends on how you prepared your library. Also will affect how well phage term works and what coverage you have. But that's kind of the best kind of established thing right now for looking at termini, aside from doing, actual empirical experiments, like restriction digests and things.
Got it. Can you upload already annotated genomes or edit and visualize annotated genomes?
Yeah, so that was covered before. So we have a tool which we're working on now, which we're to make it usable for kind of all standard files, to be able to upload into the Galaxy and have them appear as the user tracks. And so it actually appears in the user annotation track. So right now we just have there's issues with like how things are encoded, because even in GenBank it's not perfectly standardized, like what fields the annotations are in, for example, and what features are there. And so Apollo does not support all the possible features that a GenBank file can support right now, because Apollo is still being developed as well. So it can handle like a subset of those features. But we hope to have that tool available in the next month or two and test it, and so the user could upload a previously annotated genome as well. Right. So you can check on our website. We announce tools as they come out on the website, and we're trying to keep up with the Twitter also to tweet those things out.
Great. Can you use the platform to identify phage introns?
Yes. So that's one of the there's a workflow step in there which I didn't cover because that requires more time to talk about. But it's an interrupted gene finding tool. It's described in the publication that I cited at the end of the talk. And basically what it does is it searches your BLAST results for adjacent or nearby reading frames that hit like the same, an intact version in your results, and it will point those out to you. And so we've tested that quite a bit, and it's pretty good for finding intron interrupted genes, as long as there is at least one or two intact versions of that protein in the database already. And if yours is split by an intron, it will be able to find them. It also is pretty good at identifying where you have program translational frame shifts, like for example, the tape measure chaperones can also be identified by that, as long as the kind of the frame shifted extended version is annotated as a protein in the database that it can compare it to. So yeah, but we do have that functionality.
Got it. How can you download FASTA sequence from FASTQ raw files?
You mean like, well, you can convert FASTQ to FASTA. I believe there is a tool on our Galaxy to do that, and also other Galaxies too. So, you know, this is about our Galaxy, but there's the main use Galaxy, which is the big EU one. That's run on like a real kind of distributed supercomputing system, and anyone can have an account on there too, and that has a lot of standard tools. Ours has some of those tools as well. And there's a lot of these things around. So a lot of the tools that we have are just like standard packages, right? Like emboss and bed tools. And some of those things also include like standard converters, like BioPython parts. And I said, so there is a way to convert FASTQ to FASTA. It basically, you just throw out all the quality scores and then you end up with FASTA. If you're talking about assembly, then yeah, then you have to, you know, run it. You have to take your reads and run it through some kind of assembler. I mean, we use Spades for pretty much all of our assembly, but there are other assemblers out there.
Got it. Question about uploading lots of gigabytes. How can Illumina read files that are more than 2 gigabytes be uploaded to CPT Galaxy?
That's a good question. So we there is a file upload window there. There's also an FTP functionality there to upload data, but in terms of say like interfacing with base space directly, our Galaxy won't do that. We had a thing that did that for a while, but it's not really meant to do that. It was kind of a kludge, and so we got rid of that. So yeah, so if you have very large data files that are hard to transfer, then I think really you're better off to just you have to try to subsection them or trim them down. I believe we can upload larger files than 2 gigs to the system, but I'm trying to think how those happen. I think those actually might happen through the FTP system instead. So we tend not to have gigantic files because when we do our sequencing, we use a lot of indexes. So each index will only have a couple of genomes in it. So they're not like many gigabytes long. But that's something a little more technical. I'd have to look into that to see if there's a way to upload very large files into the system particularly from outside of our firewall, right? So from offsite.
Got it. How can you predict the DNA packaging strategy of a phage from its sequence?
Okay, so that's like using the phage term again. The phage term tool is pretty robust. And so that will tell you likely termini, right? But part of it's also you have to understand the biology of this. So the phage term will tell you the likely termini type and give you locations for those. You then have to reopen your genome at those points, right? And you also have to make judgment calls about whether or not you think that's correct or not, right? So our experience has been that phage term tends to, if it can't tell what the packaging site is, it will kind of just become PAC or it'll be like unknown, right? It'll tell you. In most cases where it predicts like a COS or terminal repeat, it looks like it's correct. So, but again, all that will do is just give you coordinates on your FASTA file, like where it thinks those termini are, and you have to actually interpret those results and decide if they're valid or not, in the context of your phage, and also decide what you want to do with that. Do you want to reopen your phage at a COS site even though it interrupts a gene, or like at a TR boundary if it interrupts a gene or not? If it's PAC, it's generally pretty arbitrary where you want to reopen it, and that kind of depends on what the precedence is. Like, are there other related phages? There's already a bunch of related phages in the database already that are all opened at a certain spot. Maybe you might just want to reopen your phage to be syntenic with all the rest of them. Or if it's a novel phage, you just kind of get to decide where you would like to reopen it. If it's a PAC type phage, it doesn't really have a true end, then it's kind of up to you where you would like to reopen it.
Got it. Are there de novo assembly tools for long read data from the MinION, for example, and also CANU, CANU?
For CANU, yeah. So there are tools not on our system. Those tools are very computationally intense, And so we have not put them on our system. We actually use the EU Galaxy. Use Galaxy has some of those tools. They have Canoe, and they have the other hybrid long read assembler. I think actually the new versions of Spades, I think, will also take long reads up to a point. So yeah, so those other ones, they do exist, but we don't have them on our system because they're real memory hogs. So we use actually either at the command line we use them, or we use the other Galaxy instances that offer those.
Great. Is there a way for a group to annotate a phage together, similar to how Google Docs works?
Yes. So that definitely yes. There's a tool in Galaxy, which is part of the Galaxy Apollo bridge tooling, which lets you control access from the Galaxy side. So you have an organism that you make, and it gets put into Apollo, under your account, right? So there's an Apollo account with your name on it. It goes in there. In Galaxy, there's a tool that lets you share that organism with other users, and so when they go into their Apollo, that then becomes an option, right? So there's in the Apollo, there's a drop-down menu at the top that has like all the organisms you have access to. And so if you've only annotated one phage, you only have one organism, but you can have multiple organisms in there. And then that'll become available to multiple users. You can have multiple people either working on it. It's not quite as responsive as Google Docs, right? Like, we're not Google. So it's not, you don't get like to the second updates of annotations. When you put an annotation in, it'll take a few seconds to work, and then it will, become propagated into the system. So you can't like have multiple people editing the same gene at the same time, right? So you have to kind of slow down a little bit. But definitely multiple people can have the same window open and be using be annotating the same genome at once. Yeah.
Great. Since there are other assemblers available, such as Virion2 for virus assembly, does Galaxy comprise assemblers specific to viruses only? So that's the first question.
No, so we started using Spades some years ago. We started using Velvet, and Velvet kind of went away. It's obsolete now, I think. And we've been using Spades for many years, and so we haven't really had any problems with Spades, and so that's just what we've been using for our, for general assembly. We do, we do 99% of our sequencing just using Illumina. So especially for phages, we only really use long read for if we're doing bacterial genomes, right? And that's a different ball of wax. But for phage genomes, typically we use Spades, and it usually works quite well. We haven't looked at other assemblers, although the other ones are out there. I'm sure that will also work. So I couldn't tell you about other assemblers besides that.
Yeah. Someone asks about, can you use EasyFig in Galaxy?
Yes. So we made a wrapper for EasyFig. I think we made it. Somebody made it. Might have been us. Anyway, but we do have EasyFig in our Galaxy. So that's available as a tool there. And I mean, one of the advantages of this is that if you run your own Galaxy, you can then also install EasyFig to run your own Galaxy. And as these things get popular, there's like over 100 other Galaxy instances out there, and some of them may also adopt these things. So you can use ours, and maybe there'll be other ones out there as well. But it is there now, yeah, it seems to be working. So it's an option then again. So we have a set of visualization tools there. There's a few tools that will make maps that we made, or that are derived from other mapping tools. And then we also have EasyFig. And we found EasyFig is pretty good for comparing, if they're not too different from each other and there's not too many phages, right? The thing with something like EasyFig, because it does like these multiple comparisons, if you have like a lot of phages and there's a lot of differences, you can end up with a figure that's kind of unreadable, right? Because there's just too many lines flying all over the place. So you have to think about kind of what the appropriate tool is to use for the comparison you want to make as well, so that another person can actually look at your figure and understand what's going on.
Yeah, got it. Okay, so we're getting to the end of our list. And I see we're a little past the top of the hour, so we'll just kind of hit the last couple questions, and then, yeah, this has been really awesome. So I think this question maybe you touched on already, but how there's many different Galaxy platforms out there. What Galaxy platform do you recommend for quality sequence data analysis?
for doing assembly. I think that the EU Galaxy has those tools. It has, FastQC, and I think it has Dramatic and those kinds of tools on there. So you can use that. This is what we usually refer to as like the public Galaxy, is the EU one. And so they actually do have a data retention policy though, right? So meaning if you can upload your data and you can use it. I've never maxed out my account on there, but if you come back a year later, your data is probably not going to be there anymore. So I think it saves it for a few months. Our system right now, we actually don't have a real strict data retention policy right now, but that will probably be something that we have to bring in once we have more and more users. And because we can't just store infinite terabytes of data, but users will have a lot of notice if we're going to start putting data caps or retention policies. Like, for example, user data will be stored only for 3 months or 6 months or something like that. Users will have a lot of notice about that.
Great, awesome. I think you already touched on faster versus Galaxy in the beginning.
Faster is for, so faster, that's like the, I always get confused with faster. Is faster the automated annotator, or is that for the Prophage finder?
We'll see.
Okay, they all have PH. They all start with PHs.
Yeah.
So yes, that's the Prophage one, right? So, or is faster for lifestyle prediction? Yeah. So there's a lot of these tools. So Prophage prediction is something we have as part of a workflow, right? But it's not really for analyzing bacterial genomes. You can look at your phage DNA sequence and it'll tell you what other bacterial genomes it appears in, right? If you in a prophage form, and it'll retrieve those for you. The system is not really for, say, taking a bacterial genome and telling you where all the prophages are. We don't have that. Although, with workflows and BLAST, you could actually probably put something together that would do that, something similar to that, right? And that's the power of workflows. You could say, take a bacterial genome, you'd run BLAST. We have parsers, for example, that'll take BLAST hits out and resect those regions out using a combination of our tools and things like bed tools. And then you can get those, and you could retrieve those regions from the bacterial genome. So, but we don't have, say, a specialized prophage finding tool in there. We've talked about maybe putting faster on our system, or some of the other prophage finders, because anything that runs in the command line could, in theory, be imported into Galaxy. So, for example, we've talked about putting the Phanotate, for example, right, that's one of the automated annotators. That could be put into Galaxy because it just runs the command line, and you would just, run it through there, and it would just give you the annotated genome when you're done. Proca already exists in Galaxy. I know there's a wrapper for it, although we don't have it on our system. And so those can be made, right? And then the question would just be importing it into Apollo, right? So we'd have to do some other work on that side. So there's a number of different prophage finders out there. And we've talked about importing, like having 1 or 2 of those available maybe in Galaxy. It's just how much time we have, in terms of putting it on the server. And then also, if we're the first ones to use it, then we have to write the wrapper, right? And so that takes some time to get it on there and actually have it working properly.
Awesome. Well, I think a lot of these other questions have been answered by other members of your team. So May and Ben, I see, and so that's been awesome. I'm going to share, I always share the chat log with the speakers. So I'll send that to you, Jason, if you want to see it. There's some people saying nice things like, I just want to thank you for making your platform available, new to Phage work, and I wouldn't have known where to start my annotations without it. That's from Paulina. So lots of nice things like that. So we really appreciate you taking your time. And I see May is adding some resources to the TAMU Galaxy site. So everyone who's listening, check out the chat and copy some of these links.
Yep.
And
so if you're interested,
yep.
So if you're interested in the tool, you can go to the site. So the tool window has a search at the top, right? So you can just look to see if the tool exists. And the other thing you can do is you can go to the main Galaxy, and there's a thing called the tool shed there, which is all the tools which people have made wrappers for that are available for use in Galaxy somewhere. right? And that'll tell you if anyone has ever used it in Galaxy anywhere, right? And if it's available, and you might be able to find it in there as well. But again, you know, we're still building the system out. So if there are tools that people want a lot, then, you know, that's something we can put in. So somebody mentioned that FASTR. Yeah, so FASTR, as far as I know, that's the prophage finding, and it is on a website, right? And it's a web server. So we couldn't put that on ours directly. We'd have to talk to, I think it's from the Wishart lab, FASTR. And so we'd have to talk to them to get the source code if they want to do that and make it, and it would have to be able to run on a different server, right? So if they're willing to do that, that's something that could be done. Otherwise, tools which are just straight open source, you know, they're already, if the code is just sitting there in GitHub, then that's easier to put into Galaxy.
Awesome. And we also are going to probably do a recap of this talk and include a bunch of these links and resources. So just so everybody knows, we'll publish that in our newsletter, Capsid and Tail. You can subscribe. We have a Slack channel too, where, I don't know, Jason, if you're into Slack or anyone from your team, but
yeah, we have an internal Slack that we use. So I'm not sure how to, I guess I could just sign up. There's like the Phage directory Slack also.
There's a Phage directory Slack, and you can join it. And there's a bioinformatics channel that we've been directing everyone from this bioinformatics series to go there if they have more questions. And we've been opening that up to the speakers if they want to come in and check out if there's any they can answer.
Okay, well, yeah, send me the link then. It'll be a workspace, right, in Slack.
Yeah,
so
exactly.
Yeah, send me the link to it, and I can join it.
Perfect. And yeah, it's phage.directory/Slack for anybody, but I will send it to you, Jason. Jan is posting it in the chat. That's good. Okay, yeah. So Ermi, did you have any kind of other closing remarks before we close out?
Yes, so there were several questions from YouTube viewers which were posted here. Could you take them? I think I missed a few. Could you take them all from the YouTube? Ritu was posting here on the chat. I hope they all are covered.
I have been seeing them being posted, if that's what you mean, and I've been asking some of them. Some questions I skipped if they were kind of repeats. So otherwise, if they've been here, I've been getting them.
Sure. So Jason, so what is now the percentage of genome that can be predicted with confidence, if you think about a number?
You mean like annotated?
Yeah, annotated.
You mean the number of percentage of genes that can be annotated?
Annotated in a phage genome?
It really depends on the phage. So, you know, we get, so for example, phages for Klebsiella, you know, or, you know, E. coli that are related to things like T4 or T7, you can annotate most of the genes, right. Except for you'll always have this little bunch of hypothetical proteins. But, you know, we go out to things which are more unusual, things like Vibrios or, you know, we did Colobacter some years ago. When we did Colobacter genomes, I think only about 30% of the genes had annotatable function. 70% were all hypothetical proteins. That might be a little better now. The databases have gotten better. But yeah, it's not that unusual, especially if you get a phage for an unusual host, you know, that people haven't done a lot of genomics for. It's not that unusual to have like half the proteins still be hypothetical proteins. One thing that you know, we do use sometimes for annotation that's not on the system is HHPred. And I think HHPred is getting more popular for functional annotation prediction because it's more sensitive than even an InterPro-scan. But again, it's also very computationally intensive. And so we're not offering HHPred on our system because it would probably die if somebody put, you know, 500 proteins into HHPred. Not unless we can expand our system to like a cloud infrastructure, would we be willing to do that. But right now we just use the publicly available HHPred server from Germany that everybody else uses and to search individual proteins and then just bring those back in. And that's more sensitive, but even then it's not that unusual to have a lot of, even large proteins, you put them in HHPred and they have no hits, right? So there's, I think Graham Hatfield called phages the source of the dark matter of you know, the genetic dark matter of the world. Right, and that's still true. There's still a lot of hypothetical proteins in phages.
Right. And one final question. So do you plan to apply these tools for therapy to characterize phages for therapeutic potential?
Yeah. So this is the system that we use for all of our research now. So, you know, it was developed initially for teaching. which is why actually there is quite a bit of manual intervention, but that gives you a lot more quality control as well, to have a lot of manual analysis happening. And we do use the same Galaxy system that's available to everybody. That's our main kind of research production platform. So yeah, if we have time, certainly for when we're doing therapeutics, we sequence the phages and annotate them on that platform. And one thing I didn't talk about is there's a set of workflows for doing comparative genomics as well, for finding your most related organisms. That'll be another video, that other workflow. But if you look at that computational biology paper, those workflows are all described in there.
Okay, that's awesome, really. Thank you so much, Jason, for your time. And Mai is doing the background work all this while posting the relevant links. So with this, I think we're going to close this series. I think we had a fantastic series of speakers on genome annotation, and I hope all the participants who've been here, I think, are going back far more rich in phage genome characterization, which was the intent of this series. So thank you so much. And we hope that we look forward to more of these modules that you have prepared in your series. And many people were asking in the questionnaire we asked for registration, that how can we access the software? So I think a lot of people are eager to use it, those who are not aware of it. So I hope the links that Mai has shared would reach everybody. And yeah, with that, we thank you so much, and all the participants and everybody. Ritu, also special thanks to Ritu. And of course, Jessica and Jon, always pleasure to work with you. So with this special talk, I think, close, at least for now, this series, and maybe we come up again, right? Is that okay? Is that right, Jessica?
Yeah, that sounds good. We had our four events. I think did we start in March or February? Yeah, they've all been awesome. So we're excited to make them all available, and find us in our Slack if you have trouble finding anything, recaps, recordings, and links. We're trying to get those for you. So yeah, thanks everyone for coming, and it's been so fun, Ermi. Glad we did this.
All right, well, thanks for the invitation, and good, we had a good turnout here. So so I guess I'll be in that Slack, and then you can contact us through the website. There's email addresses there, and the system is there. So
great. Well, everyone have a wonderful day / night.
All right. Okay, thanks.
Yeah. Bye.
So we may leave now. Okay.